Now let's study about prokaryotic cell. Prokaryotic cells are much smaller in size as compared to eukaryotic cells. As you can see here, if this is the size of eukaryotic cell, then this is the size of prokaryotic cells. There is no membrane-bounded organelles which is present in eukaryotic cells. If you compare prokaryotic cell to eukaryotic cell, then you will see in eukaryotic cells there are a lot of number of membrane-bounded organelles like mitochondria, Golgi apparatus, and plasmic reticulums are present in eukaryotic cells, but all these membrane-bounded organelles are absent in prokaryotic cells. Next point is no nucleus. In prokaryotic cells, there is no nucleus. So DNA is found in cytoplasm, and DNA freely floats in cytoplasm. One important thing about DNA is that DNA is not linear, it is circular. It means there is no end. And this particular DNA is known as nucleoid. DNA is not bounded to histone protein. This is also an important point for prokaryotic cells. In eukaryotic cells, you will see that DNA is bounded to histone protein and form chromosomes for packaging purpose. But there is no such thing in prokaryotic cells. Now let's come to the special additional circular DNA which is present in prokaryotic cell. Don't confuse this additional circular DNA with nucleoid. Nucleoid is separate thing. As you can see here in this diagram, let me show you this diagram. This is nucleoid, main circular DNA known as nucleoid. But this is an additional circular DNA which is known as plasmid. And this plasmid contain a special feature, means this plasmid have an gene which is resistant to antibiotics, and because of this property, this plasmid and these prokaryotic cells are used in biotechnology at very large scale. We will study more about this plasmid in biotech unit. Now let's come to the ribosome. Ribosome which is present in prokaryotic cells are 70S. S is a Swedberg unit. We will study more about this when we will study separately ribosome. And in eukaryotes, you will see that ribosome is 80S, means larger ribosome is present in eukaryotes. Now let's come to the cell wall. In prokaryotes, cell wall is made up of peptidoglycan, especially in bacteria, and also known as murin. The function of cell wall is to maintain the structure of cell and shape of the cell. Now there is two structure, flagellum and pili. Don't confuse these two things. Here you can see this one large long structure from the surface is known as flagellum, and if you look at pili, then you can see that not that much long, just small hair-like structure present on whole surface of bacteria or prokaryote, that is known as pili. Now let's see what is the function of these two things. Flagellum mainly help to move. The function of flagellum is to move of the prokaryotic cell, and pili especially help in to attach surface, or it also help to attach another bacteria, and this is important phenomena to transfer DNA. And there is one more structure which is present in prokaryotes known as mesosome. What is mesosome? Mesosome is simply enfolding of cell membrane. You can see after outermost layer is cell wall, and then a special layer is present covering cytoplasm, that is known as cell membrane. So at some point, cell membrane has enfolding structure known as mesosome, and mesosome help in respiration. So this is the prokaryotic cells, and now let's study about eukaryotic.